I want to ask you to look at these two pictures and decide what sex you would assign to each of them, male or female. And how did you make this decision? How do we actually define sex? Well, I suppose that would depend who you're asking. You know what a group of toddlers would say? Boys have short hair and girls have long hair. A psychologist would probably answer with a question. How do you feel? More of a gender approach. Trump, he would need to know if you fertilize with an egg or with a sperm. But if you ask me, well, that also depends, since I wear a few hats. I'm also a pediatrician and a clinical geneticist, but I'm also a researcher studying genetics. As a pediatrician, I'm so fortunate to see babies born all of the time. And what is the first thing you must shout in the delivery room? It's a boy or it's a girl, right? The midwife just looks down at the external genitalia and assigns boy or girl. Easy. As a researcher, a little more complicated. You see, we all start out the same, with the potential to become either male or female. But then, development continues in separate paths. These paths are determined on a few levels. The first level at fertilization, the embryo will receive two sex chromosomes, XX for females and XY for males. Later, our organs will develop. One of these organs, named the gonad, has the potential to become either ovaries or testes. These ovaries and testes will then secrete what we call sex hormones. This will further develop our reproductive system and our overall appearance to be either masculine or feminine. So we have our chromosomal sex, our gonadal sex, and our anatomical sex. Now, these usually all work in sync together and continue in a straight line. Sometimes there could be a mismatch on one of these levels. This will cause a group of disorders called differences in sex development, or DSD. And I would like to emphasize that these are true biological differences. Although used interchangeably and sometimes confusing, I'm not referring to gender identity or sexual orientation. So where do we actually meet these cases, these patients, with differences in sex development? Well, let's go back to the delivery room. Can you imagine a scenario where you cannot shout, it's a boy or it's a girl, because looking down at the external genitalia, you just can't tell? Or a teenage girl late at getting her period, under investigation finds out she doesn't have a uterus or maybe even has internal male organs. Or a couple dealing with infertility, finding out that one of them may have sex chromosomes that do not match their anatomical sex. And why do these situations occur? We know many of them can be tracked back to our DNA, our genetic material. This withholds the information of all of our features, from our eye color to diseases. And all living beings have DNA or genetic material. Our DNA is built from building blocks called nucleotides, A, T, C, and G at different combinations. But sometimes a mistake can occur, or what we would call a mutation. Now, is this a bad thing? Let's look at the word cat. If we write it with a K, it still works. But if we write it with a B, that changes the whole meaning. This is the same with our genes. Let me tell you, we all have millions of mutations. Most of them don't cause any harm, but some of them can cause DSD. So how do we know which are the good mutations and which are the bad mutations? One of the greatest achievements in human genetics is the Human Genome Project. This was a project carried out from 1990 to 2003, where a group of international scientists aimed and succeeded in sequencing our whole DNA. So now we know the order of our nucleotides, but another amazing thing that they discovered is that only around 2% of our genome actually encodes for a product. In our body, these are our proteins. Meaning, most of our DNA does not form any product. So what do we call those things that we don't have any use for? Junk. Most of our DNA is junk DNA, meaning we don't need to be focusing on it when searching for causes of diseases. And I want to show you what Clinton said when the first draft of this project was complete.
With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. Genome science will have a real impact on all our lives, and even more, on the lives of our children. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases. In coming years, doctors increasingly will be able to cure diseases like Alzheimer's, Parkinson's, diabetes, and cancer by attacking their genetic roots.
So we had it made. We can diagnose disease, cure diseases. Hey, we can even cure cancer. So tell me, why is it that over 20 years later, most of the patients with differences in sex development undergo genetic investigation and cannot find the mutation that causes their disease? To understand this, I want you to think of our genome as an orchestra. Similar to an orchestra, our genes need to be expressed at specific time, place, and intensity so that it'll all work properly. And when something goes wrong, or we have a genetic disease, a DSD, we'll be looking at the areas of the genome that encode for a product to find the cause of the disease or the mutation. In an orchestra, if the music is offbeat, we'll be looking at those who produce the music. So looking at the musicians, all talented, amazing, no problem there. What about the musical instruments? Well, they're all tuned and intact. Then what are we missing? What went wrong? Why is the orchestra offbeat? Why are patients developing DSD? And that's when we understood what we were missing. The conductor. We have 20,000 genes. Can you imagine an orchestra of 20,000 musicians playing without regulations and instructions? So who is the conductor of our genes? Where are the regulators? Back in the junkyard. And this is what I focus on in my research. I'm looking for the mutations that are causing DSD in the non-coding genome that was once called junk DNA and now known not to be such junk. Yes, I actually work in the junk. So who said you can't have all your dreams come true? But actually working in the junk is not as easy as it may seem, since we don't know exactly what we're looking for or where we're looking for. It's kind of like looking for a needle in a haystack. But what if we were looking for 2 needles, or 3 needles, or 4 needles? That would make it easier. So this is our approach with our patients. DSD is extremely rare, but if we could gather together a group of patients, that could help us focus on where to look. So we formed a collaboration, an international collaboration of undiagnosed DSD patients, although thorough genetic investigations. And we look at their non-coding DNA, looking for mutations that may explain their disease. We can further analyze these mutations with computational methods or biological methods. And when these methods aren't sufficient, we can use the assistance of mouse models. You know, mouse and human share 85% genetic resemblance, especially in our reproductive system. And anyone's ego is hurt by this, let me tell you that you're 50% banana. So back to our patients, we look at their non-coding genome to find mutations, which we can then go and model in mouse genome. Those mouse that have the same mutation or using genetic engineering, even deleting out this area, will then form DSD. This can help us explain how maybe these are the mutations that are causing the diseases in our patients. Another approach is to start with the mouse, delete out certain areas in the genome, and then finding that area that caused DSD and trying to find a match for that mutation, for those mutations in our patient cohort. So I want to end off presenting an example of a family that we are investigating. This family has 4 children. 2 of their boys have XY chromosomes, which is what we would expect. But 2 of their daughters also have XY chromosomes, meaning they should have developed as boys. So why are they female? This is a form of DSD. Looking at their coding genome, we couldn't find a reason for their disease. But then looking at their non-coding genome, these 2 sisters had mutations that were present only in their sequences and not in healthy family members or in healthy databases. We are now using additional methods, and we are very close to showing that these might be the mutations that are causing the disease in this family. So offering a diagnosis to a family is important on a few levels. The first level is understanding the biology of sex determination and development. But more importantly, now that we know what mutation we're looking for, we can offer this family healthy children without transmitting this disease on to the next generation. We can also look back at our additional DSD patients and see if these mutations can explain their disease. So I hope I've convinced you that sex is complicated. But for me, being able to combine these few hats, coming from the patients with their complicated cases, taking it back to the bench, studying them, and coming back with a diagnosis for the patients, gives me the tools to maybe make a difference for some of them. So I want to thank you very much for listening. And before we go to the break, I'd be happy to take any questions.